This big so we can get it started. Let's see here. Okay, so this set of notes is about the evolution of populations. It's chapter 23, part 1, and so I think you'll find these rather helpful with your upcoming chapter questions, which are due on Wednesday. So the first thing you need to know is just what microevolution is. So microevolution is the smallest unit of evolution as a whole. And what this is it shows changes in the allele frequencies of a population over many generations. So this is how we get such variation within singular species. So in this picture, there's a wolf and then there's several different breeds of dogs. And all of those things fall into the same general category, but they all look very, very different. Clearly, a Dalmatian looks different than a German Shepherd, which looks different than a Bulldog. But ultimately, all of those evolved from a wolf-like creature. And the reason for those differences has to do with microevolutions. So we've talked about Darwin, we talked about him yesterday, and one thing that he didn't know was how organisms were able to pass their traits on to their offspring. This was something he was unsure about, he was puzzling about, and never really came up with an answer for. However, in 1866, Mendel Gregor, the monk that we've talked about also before, published his first paper on genetics. And it was in this paper where really it was found that evolution is based on the genetic variation that exists within different traits. And so it was Mendel meeting Darwin's theories that gave us the idea that evolution is really based on genetic variation, which is so important and is something that we really need to know to totally understand how evolution works. So, where do those sources of genetic variation even come from? That's like the big question here. So, one of those are point mutations, where there's changes in one base in the DNA, which is what causes sickle cell. So, a single change in a DNA is one example. Another is chromosomal mutations, where a chromosome is deleted, duplicated, disrupted, or rearranged in some way. And these are usually quite harmful and cause very significant differences. There's also sexual recombination. And so this is what actually contributes to most of the variation within a population because it happens every time an egg and a sperm meet. And it has to do with crossing over and the recombination of genes that cause things to be very different. It's the reason why even siblings don't look exactly the same because the recombination of the parents' genes causes them to be very different and for genetic variation to arise. So, like I said, crossing over is the first time that this happens. It can happen in prophase 1 of meiosis. And then there's also independent assortment of chromosomes, which happens during the process of meiosis. And then finally, it happens during random fertilization when the egg and the sperm meet. So, population genetics, which is the focus of chapter 23 and will be the focus of most of our studies over the next about 4 days, is really the study of how populations change genetically over time. And to be able to really understand this, we have to make sure that we have a definition of a population like on point. So in this case, we're defining a population as a group of individuals that live in the same general area and can interbreed to produce fertile offspring. So a bunch of dogs, a bunch of gophers, a bunch of squirrels, a bunch of humans. What is not an example is anything that might live in the same area but can't breed to produce a fertile offspring. So a cat and a dog are different species and therefore different populations, even if they inhabit the same area. Some other definitions that we should be familiar with to be successful is the idea of a gene pool, which accounts for all of the alleles of all of the genes of all of the members of a population. So that's a lot of different pieces. So if you're breaking it down from a population, every single member, and then all of their genes, and then all of their alleles. For diploid species, there are 2 alleles for every gene, and those are either homozygous or heterozygous. The fixed allele is the idea that all members of a population have only one allele that codes for a particular trait. So the more fixed alleles there are, the lower the species diversity. So in humans, we don't have very many fixed alleles, but other species have a lot more. All of this relates to this big principle that we're going to be spending a lot of time thinking about and working with, called the Hardy-Weinberg principle. So the Hardy-Weinberg principle says that the allele and genotype frequencies of a population will remain constant from generation to generation. So this idea says that there will always be the same frequency of alleles and the same frequency of genotypes in each additional new generation within a population. However, the principle goes on to say that this is only true unless they are acted upon by forces other than Mendelian segregation and recombination. So there are some things that have to be true in order for the Hardy-Weinberg principle to be followed. In the Hardy-Weinberg principle, equilibrium is reached, and this is the idea that allele and genotype frequencies are going to remain constant. So in order for this to be true, like we said before, there has to be some factors that are in play. The first is that mutations cannot arise. The second is that there has to be random mating. There can be no natural selection occurring. The population size must be extremely large, and there cannot be gene flow. So clearly, this is not always the case in populations. However, to be able to kind of get an idea of what's going on with alleles, we can use the Hardy-Weinberg equilibrium and equation to really help us make some predictions. But in order to do that, we have to assume these 5 things are true. However, if at least one of these conditions are not met, then the population is said to be evolving, and we can't necessarily follow the Hardy-Weinberg principle. So in the Hardy-Weinberg principle, we assume that the allele frequency has genes with 2 alleles, and we call those alleles each P and Q. The P frequency is the frequency of the dominant allele, which we represent with a capital letter. And in this case, we're going to use the letter A. So the P is the dominant allele, which we'll use capital A for. The Q is the frequency of the recessive allele, which we always represent with a lowercase letter. In this case, we're using lowercase a. We always assume in the Hardy-Weinberg principle that the total of P plus Q is equal to 1. So the frequency of dominant allele plus the frequency of the recessive allele is equal to 1. It's also important to note that because of the mathematical property, we can assume that 1 minus P is equal to Q, and 1 minus Q is also equal to P. Now, using this equation, we also assume specific genotypic frequencies, which is the case that there are 3 types of genotypes. In this case, we have capital A, lowercase a, and lowercase a, lowercase a. Because we're assuming these 3 genotypic frequencies, we can also use the equation P squared plus 2PQ plus Q squared is equal to 1. Here, the P is representing the dominant allele, while the Q is representing the recessive allele. So P squared, which you see here, P squared is representing the capital A genotype. Q squared is representing the lowercase a genotype. And 2PQ is representing the capital A, lowercase a genotype. This is the details I just went through. This is an example of this idea at play. So in this Punnett square, we have two different alleles. We have CR and CW. Here, CR accounts for 64% of the population. CW accounts for 4%. And CW accounts for 16% here and 16% here. When we expand this to consider the genotypic frequencies, we say that 64% are CR from this group. Okay. 16% are CR from here and here. 4% are CW from here. And 16% are CW from here and here. So there will be many different Hardy-Weinberg problems that we'll be solving over the next few days. And so some strategies for helping you solve those are these. If you are given the genotype, so if you're given something like this here, you're going to calculate P and Q by adding up the total number of dominant and recessive alleles. If you know the phenotypes, then use the double recessive, lowercase a, to find Q squared and then Q by using this equation. And use P squared plus 2PQ plus Q squared to find the genotype frequencies if you're not given them. And finally, if P and Q are not constant from generation to generation, then the population is going to be evolving and the Hardy-Weinberg principle is no longer at play. All right, so here's an example of a practice problem. I would like you to try to solve it in your groups, and then I will talk through how to solve it. So if you want to pause the video for a minute, try to solve it, and then I'll give a brief explanation of how you would do this. All right, so to solve this problem, the first thing you're going to do is try to calculate the allele frequencies of capital A and lowercase a. To do this, you're going to just add the total number of capital A, capital A's, and half of the number of capital A, lowercase a. Because if there are 138 total alleles here, half of them are capital A, while half are lowercase a. So here I will add 1,469 to half of 138. That will give me the total number of capital A's. Similarly, to calculate the total number of lowercase a's, I would take half of 138 and add it to 5. That will give me the total number of lowercase a's. Then, using the Hardy-Weinberg equation and the tips from the previous page right here, I would go ahead and use my lowercase a number to find Q squared, and then Q, and then finally P. I'm going to go ahead and write out how I would go about solving this problem, and then if at this time you're feeling confused, have somebody go ahead and put that work out on the board, and look through it to see how it matches the work that you were able to do. Here again is another example of a Hardy-Weinberg problem. I'll let you go ahead and solve that with your group, and I will also write out the work so that it can be put over the document camera and you can check your work.